Hi, I'm Mickey, and I'm going to talk to you about computational modeling of human auditory evoked potentials. As sound enters the ear and reaches the brain, it excites several cells, and this response, this electrical response, can be measured in the far field, and this is a potential which is evoked by an auditory stimulus, an auditory evoked potential. What you see here is a very beautiful example of an auditory brainstem response, but I'm interested in more maybe examples that look more like this. And in particular, I'm interested in how these potentials or responses relate to peripheral pathology, the status of outer hair cells, inner hair cells, and auditory nerve fibers. This is non-trivial, the cochlea is complex and nonlinear, and all of these cells are mechanically coupled. So we resort to modeling. The idea with modeling is that we substitute the ear and the brain with a computational model, and if the computational model has any resemblance to the way sound is processed in the ear or the brain, we can go back and relate these responses to the pathology. So the research question here is, can we link the shape or morphology of AEPs to cochlear function, or more importantly, cochlear dysfunction? So we start with an auditory nerve model, state-of-the-art, that takes sound as an input and produces spikes in the output. It simulates a range of features of the peripheral signal processing. It was originally developed for cat and then adapted to humans or humanized. And we use a physiologically realistic implementation. We divide the cochlea into 100 segments or characteristic frequencies, CFs, and simulate up to 32,000 neurons, like in the healthy adult cochlea. And importantly, the model can be impaired. So I have control of the status of outer hair cells, inner hair cells, and auditory nerve fibers. But this is a model of the auditory nerve. It gives us spikes in the auditory nerve, and maybe we have an electrode in the mastoid, and we can use an eardrum electrode to get as close to the cochlea as possible. And we need to measure the transformation from the auditory nerve fiber to the electrode. So the cochlea is separated into two. High CFs or basal first turn, and the low and medium CFs that correspond to maybe the second and third turns, or more mid and apical neurons. And then there's a unitary response, and this is a convolution with a fixed function, so a linear transformation. And then these two are added, and we get to the predicted potential. These unitary responses are individualized, so every participant has one, and we fix them or derive them once, and then they're used for any sound stimulus. So we had two main steps. The first one is validate the model in healthy ears. Do they actually work? So we collected data from young individuals with very good hearing thresholds to a range of clicks at a range of levels with this tympanic membrane electrode, and simultaneously also a brainstem configuration from the mastoid to the forehead. These are responses in the peripheral configuration, and they're called compound action potentials to all these levels. Here we can plot the amplitudes and the latencies as a function of level of the peaks, and we can do a fitting for these lower units, so low and mid units from 100 hertz to 6 kilohertz for each participant. So what I'm showing here is the average, but we have a kernel for each participant, and we do the same at the highest level for the more basal responses. So we have these two fitting conditions, and then for the rest of the levels we can do predictions for each participant again, and you can see that these qualitatively fit quite well. We can see the stepwise pattern in the amplitudes, the latencies, the gradient fits quite well, and then despite some differences in morphology at mid-levels, this kind of worked well enough. We can also measure responses in ABRs, so this is same participants, same stimuli, same recordings, except with a different electrode position. And we do the same thing. We fit for each participant these two unitary responses or transformations for the back end of the model, and we can do the simulations, and again the simulations fit quite well. Perhaps there's some discrepancies at mid levels for the amplitudes, but this was good enough for us. We moved on. So once we've validated the model in healthy ears, we can move forward to modeling cochlear impairment. The idea here is that we have different profiles. So what I've been simulating right now would be completely unimpaired or healthy, and this corresponds to perfect hearing in terms of audiograms. But we can create different profiles of impairment based on these audiograms. You can see that I'm not changing the auditory nerve fibers because audiometry doesn't really reflect auditory nerve fiber loss. And with each of these profiles based on the audiograms, I can create on the left these auditory evoked potentials, in this case the caps at different levels. And as before, we can look at the latencies, the amplitudes, and we can also introduce a new metric, which is the cap threshold at which it can be detected. And you can see this increases with severity. These are profiles of different severities based on audiograms, but we can also look at age. So this is a histopathological human data post-mortem with different survival of different types of cells. And here we can create, same as before, profiles based on aging and look at how the responses to the caps vary in latency, amplitude, and threshold, and we can compare these profiles based on audiometry, audiograms, and age. These are all simulations to clicks, the caps. We can also look at different ones. So these are simulations to amplitude modulated tones at different levels, so we can measure or simulate EFRs and FFRs to a fixed pure tone at a high level. We can also generate a bit unrealistic profiles, so where we only change one of these knobs, for example, outer hair cell status, inner hair cell status, and auditory nerve fiber status. These are unrealistic, but they give us some information. For example, we can see that there are some differences between the hair cells amplitude response or growth functions compared to the in green auditory nerve fiber growth functions. This is reflected in the thresholds, where in blue and red the hair cells thresholds increase, whereas in green the auditory nerve fibers are quite flat. FFRs are quite robust to hair cell loss or damage, but quite sensitive to the loss of auditory nerve fibers. So we can put all of this information together in one very busy slide, where each row is a profile, different type of loss, and every column is a different electrophysiological metric. And this can provide an overview of how different damage is, according to the model, reflected in different electrophysiological or AEP metrics. So future steps, we want to use the model to simulate a large data set of rich evoked responses to different sounds with a lot of different profiles of impairment, such that we could, with this data, extract features and train a machine learning model that would classify. Then we get a patient that comes into the clinic, we measure some potentials, it goes through the model, and we can say the proportion of outer hair cells, inner hair cells, and auditory nerve fibers that this person has along the tonotopic axis, the cochlear axis. Thank you very much for listening, and hopefully we have some time for questions.